endocrine system. So as I told you on Monday, the endocrine system acts with the nervous system, integrates activities of the body and their cells. It has a large influence on metabolic activities because hormones are transported in the blood and they work on any cells that have target receptors on them. Compared to the nervous system, the endocrine system is slower to respond, but a more sustained response than we see in a typical nervous system reaction. And the endocrinology is the study of hormones in the endocrine organs. So I won't go through this table with you like we beleaguered on Monday, but it is good for you to keep in mind. You may need it when you're doing your homework. 16.1, comparing nervous and endocrine systems. So the endocrine system controls and integrates reproduction, growth and development, maintenance of electrolyte, water, nutrient balance of the blood, regulation of cellular metabolism and the energy balance, and it's also very important for mobilizing our body defenses, fight or flight responses, stress responses, for example. Exocrine glands are the other type of gland that exists. They produce non-hormonal substances like sweat or saliva. They have ducts that carry their secretions onto a membrane surface. What we're talking about in the endocrine system are the endocrine glands, which produce hormones, and they do not have the ducts. They secrete their products, and their products make their way to the bloodstream. So the endocrine glands we're going to look at primarily is the pituitary, thyroid, parathyroid, adrenal, pineal glands. We're going to talk about the hypothalamus. The hypothalamus is actually a neuroendocrine organ. It has neurotissue and endocrine tissue. We'll talk about some places that have both exocrine and endocrine functions, the pancreas, the gonads, the placenta. And then again, there are other cells and tissues and organs that produce hormones, adipose cells, thymus cells, small intestine, stomach, kidney, and heart. So we went through these on Monday, but here they are shown schematically. The endocrine system uses chemical messengers called hormones. Some of those hormones may act as autocrines, they act on the same cell that release them, or paracrines that will act on cells that are close to the ones that secrete them. Hormones are typical long-distance chemical signals traveling in the blood or the lymph. Autocrines and paracrines, while they are local chemical messengers, are not technically part of the endocrine system, but their actions will be very similar to what we talk about with hormones. Two classes of hormones are the amino acid-based hormones and the steroid-based hormones. Eicosanoids could be considered another class. It's a hormone by scientists, but usually classified as a paracrine. So for the hormones, we have amino acid-based. Those are the ones that are either modified amino acids or derivatives of amino acids, short chains of amino acids called peptides, or full-length chains called proteins. The steroids are all synthesized from cholesterol. They are the gonadal and adrenocortical hormones. They circulate systemically, but only those cells with receptors for that hormone are affected. So those cells with receptors are referred to as target cells. And when a hormone binds to its receptor, you're going to alter the activity of that target cell. Hormone action on target cells can include altering the plasma membrane permeability or membrane potential, doing so by opening and closing ion channels. It very often stimulates the synthesis of enzymes or other proteins. It can activate or deactivate enzymes. It can induce a secretory activity, or it can also stimulate mitosis, cell division. They act in one of two ways depending on the nature of the hormone, chemical nature, and the receptor location, as we discussed. So all the amino acid-based hormones, with the exception of thyroid hormone, are water-soluble hormones. They circulate in the plasma very freely, and they work on receptors found on the outside, the plasma membrane of a cell. Many of them work through G protein second messengers. They don't ever go into the cell, but they cause changes within the cell because of those second messengers that are created. The lipid-soluble hormones, which would be the steroids and thyroid hormones, go past the cell membrane, the plasma membrane, into the cytoplasm or nucleus, find their receptor there, and then activate genes directly by binding to DNA or activating DNA binding proteins. So let's take a look at those second messenger systems. We've looked at some before. The two main second messenger systems you should be aware of are the cyclic AMP, which we talked a little bit about on Monday, and the PIP2 calcium second messenger. So in the cyclic AMP signaling mechanism, the hormone binds to the receptor. That receptor activates a G protein. That G protein activates, the active G protein activates or inhibits effector enzymes such as adenylate cyclase. And adenylate cyclase takes ATP and converts it into the circular molecule CAMP or cyclic AMP. And so that cyclic AMP becomes the second messenger. And that cyclic AMP activates protein kinases that add a phosphate, so they are phosphorylating other proteins, turning them on or off. So our first messenger is the hormone, and one hormone activates a lot of second messengers, the cyclic AMP. And the cyclic AMP activates a lot of protein kinases that are flipping switches inside the cell membrane. So phosphorylated proteins, again, can either be turned on or turned off. The cyclic AMP is gotten rid of by an enzyme called phosphodiesterase, and that stops the cascade reaction. But this initial cascade has a huge amplifying effect. So again, one first messenger, one hormone binding, is going to turn on a lot of second messengers, cyclic AMP. And we've seen this schematic before. So our hormone binds to the first messenger, which activates G protein, and the G protein activates adenylate cyclase. Adenylate cyclase takes cyclic AMP and takes ATP and turns it to cyclic AMP, and then cyclic AMP activates kinases that get things, switches, moving inside the cell. The other second messenger system we didn't talk about on Monday, but is also very important, is the PIP2 calcium signaling mechanism. And in this case, the hormone activates a G protein, but the G protein turns on a different effector. It turns on phospholipase C. And when phospholipase C is turned on, it goes to a membrane protein called PIP2 and breaks it into two pieces, which are called diacylglycerol, DAG, and inositol triphosphate, IP3. Diacylglycerol activates protein kinases in much the same way that the kinases that cyclic AMP activates. Inositol triphosphate works on releasing calcium that's stored inside cellular sites, and that has a signaling effect. That calcium, in essence, becomes another second messenger. It's going to alter enzyme activity in channels. It also binds to a regulatory protein called calmodulin. And calmodulin, when bound to calcium, activates other enzymes that amplify cellular responses. Some of the other signaling mechanisms include cyclic GMP, which is second messenger for some hormones, and other hormones work without a second messenger system. Insulin is the insulin receptor is a tyrosine kinase enzyme, and it's going to put phosphates on proteins that have tyrosine in them, doing the same effect as other kinases, turning them on and turning some of them off. The lipid-soluble steroid hormones and the thyroid hormones get into the target cells and find receptors inside. And then that receptor hormone complex goes to the nucleus and binds specific regions of the DNA. And that generally initiates the transcription of DNA to messenger RNA. and that messenger RNA would be translated into proteins. Those proteins have a variety of functions, including altering metabolic activity, becoming structural proteins, or maybe even exported from the cell. So here we have a steroid hormone that diffuses through the plasma membrane, binds to its receptor, forming a receptor hormone complex, and that receptor hormone complex recognizes specific sequences of DNA and turns on the genes associated with the specific sequences. So the RNA is created, and the RNA goes to the cytoplasm and is translated into proteins. Let's see if we can get this to work.
The endocrine system consists of hormone-producing tissues located throughout the body. The circulating hormones control the rates of body processes and maintain homeostasis. Within an endocrine gland, endocrine cells secrete hormones, which diffuse into nearby capillaries. The cardiovascular system transports these hormones throughout the body, affecting target cells in various tissues. Target cells for a specific hormone have receptors that bind the unique three-dimensional shape of that hormone. Cells that lack a specific hormone receptor are unresponsive to that hormone, even if a high concentration of the hormone is present around the cells. Although cells express a variety of receptors, we'll focus on one particular class: receptors that bind water-soluble hormones and exert their effects through the intracellular second messenger, cyclic AMP. These receptors are embedded in the cell's plasma membrane because water-soluble hormones cannot enter the target cell directly. A hormone molecule, acting as the first messenger, binds to its receptor, causing the receptor to change shape. The receptor can now activate specific intracellular G proteins. The activation process begins with the release of a GDP molecule from the G protein in exchange for a GTP molecule. This causes the G protein to change shape and become active. The hormone-bound receptor activates many individual G proteins, which greatly amplifies the signal from a single hormone molecule. An activated G protein diffuses along the plasma membrane until it binds to and activates the enzyme adenylate cyclase. Once active, adenylate cyclase converts ATP to the second messenger, cyclic AMP. These many molecules of cyclic AMP from each activated cyclase enzyme represent a further amplification of the initial hormone signal. The increased concentration of cyclic AMP activates enzymes known as protein kinases. These activated protein kinases phosphorylate a variety of proteins within the cell. This changes the activity of those proteins and generates the cell's hormone response. The cell's response is characterized by the specific proteins present in the cytoplasm, only some of which can be phosphorylated by protein kinase. The cellular response diminishes rapidly once the hormone is no longer bound to its receptor. G proteins hydrolyze their bound GTP to GDP, returning to their inactive state, and dissociate from adenylate cyclase. This ends cyclic AMP production. Existing cyclic AMP is degraded by the enzyme phosphodiesterase, which is already present in the cytoplasm. As the cyclic AMP concentration returns to resting levels, protein kinase inactivates, preventing further protein phosphorylation. The target cell has now returned to its pre-stimulus condition, ready to respond to future hormone signals. The signal of a water-soluble hormone binding to a receptor on the plasma membrane is amplified through the activation of many G proteins and the generation of even more cyclic AMP. This cyclic AMP activates many protein kinases, which affect many thousands of intracellular proteins, all shaping the target cell's response. The response of many target cells produces the homeostatic and regulatory effects controlled by the endocrine system.
Okay, so then what causes the hormones to be released? A lot of it has to do with the level of hormones in the blood. They are almost all controlled by a negative feedback system. So an increased hormone effects on its target organs will inhibit further release of that hormone. That allows us, again, to keep the levels in a very narrow range, our desired range. But we have a couple of three different mechanisms for releasing hormones, including endocrine gland stimulation or nervous system modulation. The endocrine gland stimulation is in response to either humoral, neural, or hormonal stimuli. Humoral, we talked about, is changes of an ion or some other substance directly in the blood or plasma. So calcium is a good example. Low levels of calcium concentrations in the blood cause the parathyroid gland to secrete parathyroid hormone. Parathyroid hormone would then go through its actions to get calcium levels back up in the blood, and that would remove the stimulus. And that's shown here. So that's humoral. Neural stimuli, some endocrine organs have nerve connections to them, the autonomic nervous system. So sympathetic nervous system fibers can stimulate the adrenal medulla to secrete catecholamines. And hormonal stimuli means that the hormones stimulate other endocrine organs to release their hormones. Hypothalamic hormones are hormones that stimulate the release of most of the anterior pituitary hormones. Those anterior pituitary hormones stimulate targets that secrete more hormones. So we have this hypothalamic pituitary target endocrine organ feedback loop. So hormones from the final target organs will inhibit the release of anterior pituitary hormones. And that was shown here. And the nervous system can modulate each of those. It can make adjustments to hormone levels when needed, modifying stimulation or inhibition of endocrine glands. The nervous system can override normal endocrine controls under severe stress. The hypothalamic and sympathetic nervous system can override the insulin and allow blood glucose levels to increase. Again, we do have one more section to go before I break into the next section. So target cell specificity. Target cells have specific receptors that bind to the hormone. ACTH receptors are only found on cells of the adrenal cortex. Other hormone receptors are more widely distributed, such as thyroxine receptors that are on almost all cells of the body. And target cell activation will depend on levels of the hormone in the blood, the number of receptors in or on the target cell, as well as the strength of binding between the receptor and the hormone. So the amount of hormone can also influence the number of receptors of that hormone. So some hormones upregulate target cell receptors, so they make more receptors on their surface, and others downregulate. They lose receptors in response to high levels of hormones. That can help to desensitize target cells, help them to not overreact persistently to high levels of hormones. And that has to do with the half-life, the onset and the duration of hormone activity. Hormones circulate either free or bound. Steroid and thyroid have to be attached to plasma proteins. The others don't need carriers. Concentration will be a reflection of the rate of it released and the speed at which it is inactivated and or removed from the body. They can be removed by degrading enzymes of the kidneys or the liver. So the half-life would be the time required to take the level of hormone and decrease it by 1/2. That can be anywhere from a fraction of a minute to a week, depending on the hormone itself. They have different response times. Some are immediate. Some, especially the steroids, take hours to days because they are working at the level of transcription and translation. And some are inactive until they enter a target cell. The duration of a response is usually limited. It can go from 10 seconds to several hours, and the effects may disappear rapidly as blood level drops, but some do persist for hours at low blood levels. So the half-life, the onset, and duration of hormone activity will be dependent on whether the hormone is water or lipid soluble. So here, table 6.2 breaks those down for you. Interaction of hormones at target cells. The hormones can act on the same targets. Two hormones can act on the same target cell at the same time, two or more. And they can act in a number of different ways, including being permissive. So one hormone won't work unless another hormone is present. Reproductive hormones need thyroid hormone to be effective. Another is synergism. One hormone produces the same effect on a target cell, so they amplify the response. So both glucagon and epinephrine cause the liver to release glucose. And antagonism. In this case, one or more hormones opposes the action of another hormone. So we know that insulin is going to help get sugar out of the blood, while glucagon helps get sugar into the blood. So they are antagonists of each other. And now I am really going to stop. This is where I want to separate 16.6 on its own.